Assalamualaikum.
And hi,
My name is Nurul Ain.
and my name is Siti Zulaika.
Today we want to discuss about topic 1, biodiversity.
We want to discuss about topic 1.2, domain bacteria and domain archaea. So for learning outcomes, A. State 2 domain of prokaryotes, example E. coli, and archaea, example sulfolobus. B. Differentiate between the two domain of prokaryotes, bacteria, example E. coli, and archaea, example sulfolobus, based on C3. 3. Cell wall structure, association of histone to DNA, and structure of membrane lipids.
For C, describe the diversity of bacteria. For example, cell shape and gram stain. D, explain the roles of bacteria. 1. Recycling of chemical element in ecosystem. For example, Rhizobium sp. Second, symbiotic. For example, E. coli. Third, pathogenic. For example, Salmonella sp. And in research and technology, for example, bacterial plasmid.
Okay, so for two fundamentally different groups of prokaryotes, that is domain archaea and domain bacteria. So for prokaryotes are unicellular. Genetic material are single cellular DNA located in nuclear region. Some bacteria and archaea has extracellular DNA known as plasmid. So for domain archaea were originally discovered living in extreme environmental conditions. Three main types of archaea are still distinguished based on their unique habitats, that is methanogens, halophiles, and thermoacidophiles. We want to discuss about classification of prokaryotes. So, prokaryotes are divided into two domains, that is domain archaea, for example, sulfolobus, and domain bacteria, for example, E. coli. Okay, so we want to differentiate between the two domain of prokaryotes, that is domain bacteria and archaea. So for domain bacteria, cell wall contain peptidoglycan. For archaea, cell wall contain polysaccharide and protein, lack of peptidoglycan. So next is domain bacteria, DNA is not associated with histone protein, while archaea, DNA is associated with histone protein. So lastly, domain bacteria, lipids in plasma membrane is unbranched. Hydrocarbons bonded to glycerol by ester linkage. So for archaea, lipid in plasma membrane is branched. Hydrocarbons bonded to glycerol by ester linkages.
I want to discuss about cell shape. Cell shape have 4 shape, which is spherical shape, rod shape, spiral shape, and coma shape. For spherical shape, known as coccus, they have unicellular, 2 cell, and colonized. For example, Diplococcus sp. Next, rod shape. Rod shape, known as bacillus, they unicellular and colonized. For example, E. coli. Next, spiral shape, known as spirillum, unicellular. For example, Rhodospirillum sp. Lastly, coma shape. Coma shape, known as vibro, unicellular. For example, Vibro cholera. Okay, we now to discuss about gram stain. Gram staining is a tool for identifying specific bacteria based on the difference in their cell wall. Gram staining have two, which is gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria has simple cell wall, thick peptidoglycan layer, stain blue or purple, crystal violet, trapped in the peptidoglycan, less pathogenic. Gram-negative bacteria has more complex cell wall, consists of outer membrane that consists lipopolysaccharide thin peptidoglycan layer. Outer membrane protect them from the host defense. For example, Salmonella sp. Okay, now we want to discuss about difference between gram-positive and gram-negative bacteria. Gram-positive, they have cell wall has one layer, thick peptidoglycan. Gram-negative, cell wall has two layers, thin peptidoglycan and outer membrane. Okay. Gram-positive, staining with crystal violet will return purple color, while gram-negative, staining with crystal violet does not return purple color, stain pink from safranin. Gram-positive less pathogenic, while gram-negative more pathogenic. That's all.
Thank you.
Thank you.